We'll be discussing today on the nucleic acid extraction methods, particularly DNA isolation. The objective of this lecture is to discuss how DNA is isolated from various biologic specimens. We also be discussing the principles for handling of all clinical specimens for DNA isolation. We'd also compare and contrast organic, inorganic, and solid phase approaches for isolating cellular and mitochondrial DNA, and we'll be describing how DNA isolation from minimal and challenging samples. But the purpose of nucleic acid extraction is to release the nucleic acids from the cell for use in subsequent procedures. Now remember, our nucleic acids, particularly DNA, it is located inside the nucleus. And our goal for nucleic acid extraction is to isolate that nucleic acid in order for us to study, for example, in DNA sequencing or in gel electrophoresis. Ideally, the target nucleic acid should be free of contamination with protein. Now remember, DNA, inside our nucleus, the DNA is organized into a chromatin. And chromatin is not just a bunch of DNA. It also contains a lot of proteins, examples of which are the histones, which makes up the nucleosomes. So we want that sample to be free of protein, of any carbohydrates, lipids, and other nucleic acid. That is DNA free of RNA or RNA free of DNA. The initial release of the cellular material is achieved by breaking the cell wall if present, for example, in fungi or bacteria or in plants or algae, because they are the organisms that contain cell wall, and the cell and the nuclear membrane through cell lysis. So we want to break the barriers that contain the nucleic acids. Lysis must take place in conditions that will not damage the nucleic acids, because if we lyse the nuclear membrane and also at the same time we damage the nucleic acids, now we will get a sample that is also lysed. The DNA is also lysed. The DNA is also already broken down. So that is useless. Now, following lysis, the target material is purified, and then the concentration and purity of the sample can be determined. So the following are the principles of handling for all clinical specimens. We should observe universal precautions for biohazards, especially if we are handling microorganisms which are of potentially damaging or potentially hazardous to the community. We should also use protective gowns, gloves, face, and eye shields when we handle clinical specimen for DNA isolation. We should also decontaminate all spills and work areas with 10% bleach, which is sodium hypochlorite. We must also dispose of all waste in appropriate biological waste containers. And of course, we should use gloves. The DNA extraction is the isolation and purification of DNA or deoxyribonucleic acid. The sources of DNA would include plasmid DNA for those microorganisms such as bacteria, viral nucleic acids because some viruses have DNA as their genome. Examples are the papilloma viruses, hepatitis B viruses. We also have the adenoviruses and the herpes viruses. They have a DNA as their genome. Genomic DNA from blood and biological fluids are also sources of DNA. From tissue and cells, from forensic samples, from plant and fungi, from food and feeds. Basically, anything that may contain a nucleic acid can be a potential source of DNA. The purpose of DNA extraction is to obtain DNA in a relatively purified. Okay, the highlight of this one is to get a purified sample of DNA. And we say purified, it's pure DNA, nothing more. Nothing more of it. No protein, no carbohydrate, no lipids, from which can be used for further investigation, such as PCR or polymerase chain reaction, for restriction fragment length polymorphism analysis or RFLP, and for Southern blotting. The types of specimen for molecular diagnostic laboratory, especially in medicine, we have the whole blood, because blood contains a lot of formed elements. So remember, whole blood the two major components of blood is plasma and formed elements. Now in plasma, it's mostly water, ions, proteins. So you wouldn't expect any nucleic acids from there. But the formed elements, we have various formed elements. The three main formed elements are the erythrocytes, the leukocytes, and the thrombocytes. Now, among the three, you wouldn't expect any nucleic acids from erythrocytes. Why? Because erythrocytes are enucleate, meaning they do not have any nucleus, meaning they do not have any DNA. So, the source of DNA from whole blood would most likely come from the leukocytes, which are the neutrophils, basophils, eosinophils, monocytes, and the lymphocytes. The thrombocytes, they are just fragments of your megakaryocytes. So you wouldn't also expect nucleic acids from there. Okay, PBSC is a phoresis product, serum or plasma. Okay, especially those if we are considering viral infections or bacterial infections that are present in blood, we can also get it from buccal cells, from cultured cells, blood spots, body fluids such as CSF or the cerebrospinal fluid, bronchoalveolar lavage, from amniotic fluid, from semen, and urine. Tissue samples would include a fresh tissue sample or a frozen tissue sample, or can be paraffin-embedded tissue sample. And also, hair can be a source of specimen for molecular diagnostic laboratory, especially if you have the root. Okay, you wouldn't expect any nucleic acid in the hair itself, but you can find it in the root of the hair. Okay. In specimen labeling, we should always include the patient name, date of birth, and medical record number. The ordering physician, the type of specimen, the accession number, date and time of collection, the laboratory technician identification, or even just the initials, and the requested tests. The DNA extraction is also used to isolate mitochondrial DNA and genomic DNA. And DNA can be extracted from cells or tissues, environmental samples, or any nucleated cells. Now, it's not just the nucleus that contains DNA. Mitochondria also contains DNA as well as chloroplasts in plants and algae. Preparing the sample, the nucleic acid is routinely isolated from fungal, bacterial, viral, and human sources in the clinical laboratory. The initial steps in nucleic acid isolation depends on the nature of the starting material and the test method. Some test systems include sample collection vessels and reagents that begin the nucleic isolation and transport to the laboratory. Details of sample collection are important because of the diversity of sample types and nucleic acid sources such as viruses, bacteria, and nucleated cells. Use of an improper collection tube or method will compromise the test results. Test validation should include sample collection methods. The yield of DNA from different specimen sources also varies from one another. As you can see here in table 3.1, we see that in blood, the expected yield, especially we have the blood in the buffy coat, we have an expected yield of 50 to 200 microgram of DNA. The bone marrow, we are expecting to have 100 to 500 micrograms. For cultured cells, we have 30 to 70 micrograms. For solid tissue, we have 1 to 10 micrograms. For lavage fluids such as bronchoalveolar lavage, we have 2 to 250 micrograms. For the mitochondria, we have 1 to 10 micrograms. For plasmid DNA and bacterial culture, 350 microgram to 1 milligram. For bacterial culture with a 0.5 ml specimen and 0.7 absorbance units, we are expecting to have 10 to 35 micrograms of DNA. For feces, of which is 1 milligram of sample, which may contain bacteria or fungi, we are expecting to have 2 to 228 micrograms of DNA. Okay. For blood and bone marrow, the isolation of nucleic acid would depend on isolating the genomic DNA and RNA. And in the collection of blood and bone marrow, we collect it in an anticoagulant, in a container with an anticoagulant. We mix it well but gently to avoid disruption of cells. And the anticoagulants that we usually use are the EDTA, which are the lavender top vacutainer, and which is the preferred specimen. For ACD, these are usually the yellow top vacutainer. For heparin, we have the green top vacutainer, because this heparin inhibits several enzymes used in molecular assays. Okay. Very important also in blood and bone marrow specimen collection is that you should not freeze the specimen, because freezing the specimen will damage the membranes and eventually damage the nucleic acids. We also need to protect it from temperature extremes, not too high and not too low. Overnight delivery is preferred, and packaging must comply with the shipping rules for blood-borne pathogens. It should be sealed in a protective container, an absorbent material in packing, with a sealed container in plastic bag, and it should be labeled as biohazard. The effect of tissue fixative on purification nucleic acids are the following. So we have various fixatives, such as the neutral buffered formalin, B5 fixative, Bowen's fixative, Zenker's fixative, and alcohol. For neutral buffered formalin, the active component is formaldehyde, and the tissue effect is nucleic acid-based hydroxymethylation and cross-linking of DNA and protein. And what is the effect in the nucleic acid purification? You would have reduced high molecular weight nucleic acid with increased fixation time. And this is suitable for most testing. For B5 fixative, the active components are mercury chloride, and that is usually mixed with formaldehyde. The tissue effect is that we have mercury protein complexes that reduce DNA extraction yields. And the effect of nucleic acid purification is that it would result into a low molecular weight or no extractable nucleic acid. Okay, occasionally, nucleic acid sufficient for PCR testing. For Bowen's fixative, the active components are picric acid, acetic acid, and formaldehyde. The tissue effect of Bowen's fixative is that it acidifies DNA depurination, and also the effects of formaldehyde, which are nucleic acid-based hydroxymethylation and cross-linking of DNA and protein. The effect of its purification, you would have low molecular weight or no extractable nucleic acid. For Zenker's fixative, we have potassium dichromate and mercury chloride. The tissue effect is we have formation of metal protein complexes. And still the same as that of Bowen's fixative, it can result into no extractable nucleic acid. Okay. For alcohol, we have ethanol or methanol. The effect of its nucleic acid purification is that it can be a good or excellent, it can have a good or excellent nucleic acid yields, including high molecular weight. And fixation time has no effect. For paraffin-embedded tissue section, it is usually used in genetic testing, infectious disease testing, and identity testing. And formalin-fixed tissue is suitable. Mercury or other heavy metal fixative are not acceptable. Tissue sections on glass slides can be used for in situ application and microdissection techniques. The specimen storage requirements for DNA for blood, from samples from blood, bone marrows, and other fluids is as follows. For 22 to 25 degrees Celsius is not recommended for less than 24 hours. For 2 to 8 degrees Celsius, it is a suitable condition up to 72 hours. Negative 20 degrees Celsius is not recommended. Do not freeze blood or bone marrow before lysing red blood cells, but the leukocyte pellet can be frozen for up to 1 year. Negative 70 degrees Celsius is not recommended. Okay, and this is the storage of DNA specimen. So as you can see here, it is not recommended for you to freeze in low temperature. Okay, especially in low temperature. So if you are if you'll be using less than 4 months, then the temperature is around 2 to 25 degrees Celsius. For 1 to 3 years, should be stored at 2 to 8 degrees Celsius. For less than 7 years, should be negative 20 degrees Celsius. For more than 7 years, at negative 70 degrees Celsius. And this is recommended for long-term storage in ethanol. What is the size or volume of each sample? Amount of DNA or RNA is required, and equipment and test tube sizes are required. So we need to specify that before considering nucleic acid amplification or nucleic acid isolation. How many samples are being processed? And this depends on the capacity of the centrifuge and isolation method speed. Is a high throughput or automated system available? Now, you should also take into consideration if you have a 96 well plate method or a walkaway or semi-automation method, then you can use this automated system method. So, what are the factors that you would consider in choosing an isolation method? So the important factors include the processing speed, the ease of use, yield of DNA or RNA, quality of DNA and RNA prepared, or amplification performance, the shelf life or storage condition, quality assurance criteria, and the cost of preparation. Now, this slide shows you the basic steps in isolating DNA from clinical specimen. Okay? For example, if it's from the blood, we separate white blood cells from red blood cells if necessary. Since red blood cells are considered to be enucleate, meaning they do not have a nucleus, we want to separate them from WBC. Okay? Then after separating the white blood cells from the red blood cells, we lyse white blood cells from other nucleated cells. Next is to denature or digest proteins. Then we need to separate the contaminants such as protein, heme from the DNA. Then we precipitate DNA if necessary, and then resuspend DNA in a final buffer. DNA isolation methods can be of two major types. We have chemical DNA extraction method and physical DNA extraction method. For chemical DNA extraction method, we have also two types. We have organic DNA extraction methods and inorganic DNA extraction method. For DNA extraction methods, we have the phenol chloroform DNA extraction method. For inorganic DNA extraction methods, we have proteinase K DNA extraction method, we have the salting out method, and the silica gel-based DNA extraction method. For physical extraction methods, we have magnetic bead DNA extraction and paper DNA extraction method. For the DNA isolation method, in terms of the first chemical method, we have the liquid phase organic extraction. This is perhaps the most basic of all procedures in molecular biology. And this is purification of DNA by organic extraction. This is an organic extraction because we utilize phenol and chloroform, which are organic substances. So we have phenol and chloroform, our isomeryl alcohol, as our solution. The lyse samples mixed with this one, and the two layers are formed. The proteins remain at the interface, and DNA is removed at the top aqueous layer. And the DNA is precipitated with alcohol and rehydrated. The disadvantage of the liquid phase organic extraction method is that it is slow, labor intensive, and it includes toxic substances such as phenol and chloroform. And it requires fume hood and disposal of hazard materials. So this is the usual method of organic extraction method. Now, after the lysis of cells, the cell contents are acidified and particulate matter is pelleted and extracted with phenol and chloroform. So first we have cells in suspension. Then we undergo lysis to break the membranes or the cell wall if it contains a cell wall. Okay, the lysed cells will be acidified, and the acidification would include acetic acid and salt. Okay, now we have, after acidification, there will be a precipitation of cell debris. Okay. Next is the aqueous solution here, this one. This contains the DNA that can be precipitated by mixing with ethanol. Okay, so phenol or chloroform is used also for extraction of the DNA. Okay, and after adding phenol or chloroform, ethanol is added. And once ethanol is added, the DNA will be precipitated at the bottom of the tube. Okay, collecting the precipitated DNA by centrifugation and resuspending it in a small volume of water or vapor increases the DNA concentration of the isolated DNA. The cell lysis buffer will include non-ionic detergents such as SDS and tris chloride, EDTA, which are designed to lyse out the outer membrane and nuclear membrane. So remember here, in the lysis process, we can use these substances to lyse the membranes. The EDTA, or the ethylene diamine tetraacetic acid disodium salt, is a chelating agent of divalent cations such as magnesium. And magnesium is a cofactor for DNA's nucleases. And if the magnesium is bound up by EDTA, nucleases are inactivated. So what does the detergent do? Remember that each cell is surrounded by a cell membrane, and DNA is in the nucleus of a cell, which is also surrounded by a membrane. Okay? The membranes must be broken open in order to get out of the DNA. So the detergents, functions of the detergents, that they break apart the membranes by attaching to the lipids or fats and proteins and the membrane. So the detergent contains a polar and nonpolar region. If it interacts with the membrane, it causes destruction of the cell membrane. The cell and nuclear membranes have been broken apart, as well as of all the organelle membranes, such as those around the mitochondria and chloroplast. So what is left? Now we are left with proteins, carbohydrates, and DNA. Now we have proteinase K, which is one of the reagents, and it is usual to remove most of the protein by digesting it with proteolytic enzymes such as pronase or proteinase K, which are active against broad spectrum of native proteins before extracting with organic solvent. Proteinase K is approximately tenfold more active on denatured proteins, and proteins can be denatured by SDS or by heat. The next reagent for organic extraction is the phenol or chloroform. The standard way to remove proteins from nucleic acid solution is to extract once with phenol, once with 1:1 mixture of phenol and chloroform, and once with chloroform. This procedure takes advantage of the fact that deproteinization is more efficient when two different organic solvents are used instead of one. The final extraction of chloroform removes any lingering traces of phenol from the nucleic acid preparation. So what is the purpose of adding the chloroform in the last portion? It removes the phenol in the initial. Phenol is highly corrosive and can cause severe burns. The phenol often means phenol equilibrated with buffers such as TE and contains 0.1 hydroxyquinoline and 0.2 B-mercaptoethanol, which is added as an antioxidant. The hydroxyquinoline gives the phenol a yellow color, making it easier to identify the phases or the layer. The chloroform often means 24:1 volume per volume mixture of chloroform and isoamyl alcohol. The isoamyl alcohol is added to help prevent foaming. So the phenol chloroform isoamyl alcohol ratio is 25:24:1. What's the purpose of the phenol? The phenol denatures protein and dissolves denatured proteins. Chloroform is also a protein denaturant, but the role also of the chloroform is to remove excess phenol. The nucleases can also be added to remove unwanted DNA or RNA. Okay? If we want to isolate RNA, then we need to add DNAs to remove the DNA. If we want to isolate DNA, we can add RNAs to remove their RNA. So it depending on when nucleases treatment it is performed, it may be necessary to repeat purification steps for protein removal with the use of phenyl chloroform isoamyl extraction method. The most widely used method for concentrating DNA is precipitation with ethanol. Now we have the DNA that has already been purified. Now, how do we get a hold of that DNA? So the one way of getting a hold of that DNA is to precipitate the DNA. Now remember, DNA is in the solution. So what we want is that we want a sample of the DNA that is precipitated at the bottom of the test tube. So ethanol is a good way of concentrating DNA or to precipitate DNA. The precipitate of nucleic acid forms in the presence of moderate concentrations of monovalent cations, such as salt containing sodium, and it is recovered by centrifugation and is redissolved in appropriate buffers such as TE. The technique is rapid and is quantitative, even with nanogram amounts of DNA. The salts interrupt hydrogen bonds between water and DNA molecules. That's the function of adding the salt. And the DNA is then precipitated from the protein with subsequent step with isopropanol or ethanol. In the presence of cations, Ethanol induces a structural change in DNA molecules that causes them to aggregate and precipitate out of the solution. So before adding ethanol, the DNA is suspended in this mixture. But after adding ethanol, it creates the precipitation of DNA. And remember that this solution must contain salt because the salt will disrupt the hydrogen bonds from the water and the DNA. The four critical variables are the purity of the DNA, its molecular weight, its concentration, and the speed at which it is pelleted. DNA with concentration as low as 20 nanograms per ml will form a precipitate that can be quantitatively recovered. Typically, 2 volumes of ice-cold ethanol are added to precipitated DNA, and very short DNA molecules are precipitated inefficiently by alcohol. The optimum pelleting condition depends on the DNA concentration, and relatively vigorous microcentrifuge steps such as 15 minutes at or below room temperature at 12,000 rotations per minute are designed to minimize the loss of DNA from samples with yields in the range of a few micrograms or less. Solutes that may be trapped in the precipitate may be removed by washing the DNA pellet in a solution of 70% ethanol. To make certain that no DNA is lost during washing, added 70% ethanol until the tube is two-thirds is full. Then we recentrifuge the solution, and after the 70% ethanol wash, the pellet does not adhere well to the tube, so great care must be taken when removing the supernatant. Isopropanol with 1 volume may be used in place of ethanol to precipitate DNA, and precipitation with isopropanol has the advantage that the volume of liquid to be centrifuged is small. Isopropanol is less volatile than ethanol, and it is more difficult to remove the last tracers. Moreover, solutes such as sodium chloride are more easily co-precipitated with DNA when isopropanol is used. So before adding alcohol, causes the precipitation of the nucleic acid. Then after adding alcohol, we need to centrifuge it so that we can separate the supernatant from the pellet. Okay, the pellet is the one that contains the nucleic acid. Okay, wash the pellet with 70% ethanol to remove residual salts and other contaminants. Then we centrifuge it again, and we discard the ethanol. Okay, and allow the pellet to dry. Okay, now the pellet will be dissolved in water or in buffers such as TE. So again, we need to add alcohol because the alcohol will cause precipitation of DNA. And we need to wash it again with alcohol. And we recentrifuge it again. And then now we have the final product. Now let's move on to the inorganic extraction method. Cell membranes are lysed and proteins are denatured by detergents such as SDS. And RNA is removed by RNAs. Proteins are precipitated with salt solution, and DNA is precipitated with alcohol and rehydrated. The advantages of the inorganic extraction method is that it is fast and easy. It uses non-toxic materials, no fume hood required, and no hazardous materials disposal and no disposal issues. It produces high quality DNA. So this is the basic steps of the inorganic extraction method. So we have cells in suspension, then we undergo lysis using TRIS, EDTA, or SDS. The lysed cells contain both protein, DNA, and carbohydrates. The protein precipitation will be will be carried out using sodium acetate, okay, and this will cause protein precipitation, and the DNA is left in the solution, in this aqueous solution, and it is transferred to another tube, okay. Then the supernatant containing the DNA is mixed with ethanol or isopropanol to precipitate DNA and to be collected and suspended in a smaller volume. Now, in contrast to the organic method, it doesn't use any phenol, chloroform, or isoamine. Next, we have the solid phase procedure. For solid phase procedures, uses solid support columns, magnetic beads, or chelating agents. Support solid columns are fibrous or silica materials that bind DNA, allowing separation from other contaminants. Magnetic beads, the action of the magnetic beads that it binds to DNA, and the beads are separated from other contaminants with a magnet. Chelating resins are also used. The advantages of a solid phase procedure is that it is fast and easy and no precipitation required. So what happens in solid phase procedure? First, we have cells in suspension. Lysis occurs. So we have now the lysed cells. Okay. Now the DNA, before applying to the silicate column, particulates from the cell debris may be removed by centrifugation. And solid phase system include proprietary buffers and solutions for adhering the DNA on column, washing it and eluting it in a small volumes, around 10 to 100 microliter. So this column, the surface of this column contains beads that in which that it can bind DNA. Solid phase isolation is the methodology employed for most robotic DNA isolation system that used magnetized glass beads or membranes that bind DNA. These systems are finding increased use in clinical laboratories for the automated isolation of DNA from cultures, blood, tissue, bone marrow, plasma, and other body fluids. DNA isolated from specimens such as urine and amniotic fluid has been reported to work better in PCR application, possibly due to the removal of the inhibitory substances found in these types of specimen by the pre-filtering step. In most systems, a measured amount of sample, for example, 200 to 1,000 microliters of blood or 100 microliter cell suspension containing 106 to 107 cells or 10 to 50 milligrams of tissue in tissue samples is placed into the instrument along with the cartridges or racks of tubes containing the reagents for solution. Reagents are formulated in sets depending on the type and amount of starting material. The instrument is then reprogrammed to lyse the cell and isolate and elute the DNA automatically. Next, we have the isolation of mitochondrial DNA. There are two approaches to the isolation of mitochondrial DNA from eukaryotic cell. One method is to isolate the mitochondria by centrifugation. After preparations are homogenized by grinding on ice, the homogenate is centrifuged at low speed, around 700 to 2,600 times G, to pellet in intact cells, nuclei, and cell debris. The mitochondria are pelleted from the supernatant in a second high-speed centrifugation, around 10,000 to 16,000 times, and are lysed with the detergent. So the first centrifugation will remove the intact cell, the nucleus, and cellular debris. The mitochondria will be present in the supernatant. Then the supernatant will undergo again another cycle of centrifugation, but this time it's a higher speed. The lysate is treated with proteinase to remove protein contaminants, and mitochondrial DNA can be precipitated with cold ethanol and resuspended in water or appropriate buffers for analysis. The second approach to mitochondrial DNA is the preparation is to isolate the total DNA as described previously. The preparation will contain mitochondrial DNA that can be analyzed within the total DNA background by hybridization or PCR. Okay, so in comparison, we have the liquid phase and solid phase of DNA. So this is the summary. For the liquid phase, we lyse the RBC, we lyse the cell, we have protein digestion with the use of proteinase K, separate proteins for DNA, and precipitate DNA alcohol, and then we rehydrate the DNA using buffers. For solid phase DNA, we pre-lyse the cell, apply sample, wash the DNA, and then elute the DNA. We have now the rapid extraction methods, and with the advent of polymerase chain reaction, new and faster methods of DNA isolation have been developed. The minimal sample requirements for amplification procedures allow for the use of material previously not utilizable for analysis. Rapid lysis method and DNA extraction or storage card provide sufficiently clean DNA that can be used for amplification. Chelex is a cation chelating resin that can be used for simple extraction of DNA from minimal samples. A suspension of 10% Chelex resin beads is mixed with the specimen, and the cells are lysed by boiling. After centrifugation of the suspension, DNA in the supernatant is cooled and may be further extracted with chloroform before use in amplification procedure. This method is most commonly used for forensic applications, but also be used for purification of DNA from clinical samples and fixed paraffin-embedded specimen and samples spotted on storage cards. So this is the step for Chelex extraction. The sample is suspended in 5% Chelex and water is incubated at 56 degrees Celsius for 30 minutes. The sample is boiled to lyse the cells and destroy protein. The sample is then centrifuged and the supernatant contains the DNA and can be directly used for PCR. So that ends the lecture on DNA isolation methods. Thank you for your time and listening.